A single medical treatment today can carry a price tag of $3.5 million. This staggering figure represents the cost of our first real steps into the world of gene therapy, specifically for conditions like hemophilia B. But while the financial cost is immense, the ethical price of this new frontier became even more apparent in 2018. That year, a researcher in China shocked the global community by announcing the birth of the world's first genetically edited twin babies. This act bypassed nearly every international safety and ethical guideline, signaling that we have officially entered an era where the code of life is no longer a fixed script, but a document we can actively rewrite. The question for all of us now is whether we have the collective wisdom to handle these genetic scissors. At its simplest, the technology known as CRISPR-associated protein 9 is a naturally occurring defense system found in bacteria. Scientists have repurposed it into a tool that can add, remove, or alter DNA sequences with a level of precision that was previously impossible. However, there is a fundamental red line in this field that defines the entire debate. On one side is somatic genome editing, which targets non-reproductive cells like skin or blood. These changes only affect the individual patient and are currently used to treat devastating diseases like sickle cell anemia. On the other side is heritable human genome editing, often called germline editing. This involves altering the DNA of embryos or reproductive cells, meaning every change made is passed down to all future generations. While somatic editing is widely embraced as a medical breakthrough, germline editing is seen by many as a dangerous leap into unpredictable biological territory. This capability didn't appear overnight. It evolved from a specialized bacterial immunity tool into the primary driver of a global biotechnological revolution. Way back in 1997, long before the current explosion of interest, the United Nations Educational, Scientific and Cultural Organization, implying we have a collective responsibility to protect it. By 2012, researchers Emmanuelle Charpentier and Jennifer Doudna developed the high-efficiency method that eventually earned them the 2020 Nobel Prize. The 2018 CRISPR babies controversy served as a global wake-up call, forcing major bodies like the World Health Organization and the National Academies of Sciences to establish formal governance frameworks to prevent rogue experimentation. The debate today is a clash of deeply held human perspectives. From a medical standpoint, the promise is breathtaking. In late 2023, the Food and Drug Administration approved Casgee, the first therapy of this kind for sickle cell disease, marking a historic milestone for patients who previously had few options. However, the human rights perspective remains more cautious. The Oviedo Convention is currently the only legally binding international instrument that prohibits germline editing, arguing that it could jeopardize the inherent dignity of human beings and potentially revive eugenic ideologies. Social justice advocates add another vital layer to this conversation. Bioethicists warn that if these therapies are only available to the wealthy, we risk creating a biological caste system. This concern is compounded by a history of medical exclusion. For example, a 2016 analysis found that a staggering 96% of participants in genome-wide association studies were of European ancestry. This means that the tools being developed might be less effective or even unsafe for the minority populations who need them most. Even our environment is at stake. Scientists are exploring gene drives to bypass traditional inheritance and potentially eradicate malaria by making mosquitoes infertile. However, the national academies warned that these changes are impossible to completely roll back once released and could have unintended ecological consequences. The consequence of having inconsistent global regulations is the rise of ethics tourism. This is a phenomenon where patients or researchers seek unproven genetic interventions in jurisdictions with weak oversight. To combat this, the World Health Organization has implemented a human genome editing registry to track all clinical trials and ensure transparency. In the United States, the Food and Drug Administration has issued strict roadmaps for somatic therapies, requiring extensive data to catch off-target edits, unintended mutations elsewhere in the genome. To bridge the social gap, many experts are advocating for community-based participative research. This approach treats minority groups as informed power brokers rather than just subjects, ensuring that genetic progress benefits everyone, not just the most advantaged. As we move forward, we are witnessing the convergence of this technology with artificial intelligence. Algorithms are now being used to predict genetic outcomes and even design novel protein sequences. This means the technology is moving faster than our laws can keep up. We are no longer just observing evolution, we are actively directing it. Our shared biological heritage is now in our own hands. The question for our generation is no longer just can we edit the code of life, but should we, and for whose benefit?